Welcome to the webinar Delft3D Phytoplankton Modeling Concepts of Bloom. This webinar is broadcast to you by Deltaris Academy. My name is Edward Melger and I'm the Deltaris Academy Program Manager and also the Delft3D Product Manager. Today I have the pleasure to give a short introduction and to announce the webinar speaker. First, a few facts and figures. In total, more than 700 water quality and ecology experts worldwide have showed interest. 275 participants from 50 countries worldwide have signed in for this webinar, from Australia, Brazil, China, Germany, Singapore and United States. Again, welcome to you all. During this webinar, we have a panel of experts available to answer the questions received via text in the GoToWebinar environment. After the webinar presentation, there is an opportunity to put your verbal questions to today's speaker. About today's speaker. He's a specialist researcher and expert advisor with almost 40 years of experience in ecological and water quality issues. He has been involved in numerous studies, often as a project leader, scientific coordinator, or scientific advisor. His experience covers a vast range of both fresh and marine waters, shallow and deep, floating and stagnant, stratified and mixed. These include over 50 different lakes and reservoirs, rivers, estuaries and coastal seas worldwide. And now, give the webinar floor to Dr. Hans Loss of Deltaris.
Okay, a warm welcome to all of you attending this webinar on Delft3D Phytoplankton Modeling Concepts of Bloom. I will start with a very short overview of all the issues that will be dealt with during this webinar. First of all, I'll tell you something about the role of primary producers, then about the position of Bloom inside the Delft3D modeling suite. I'll give an overview of the concept of Bloom and then I'll zoom in more detail one of its most important principles, competition. I'll tell something about the existing knowledge base, show you some validation results, and I'll end up with a summary and advice for further reading. Everyone probably has some image about aquatic ecosystems, something like this. There's a lot of things happening and it looks perhaps a bit chaotic, but actually there's a lot of structure in it. First of all, there are physical processes. There is exchange with the atmosphere, sedimentation, resuspension, horizontal water movement, possibly stratification, and not shown here, there's seasonality. Then next is the food web, so the biological part, all the arrows pointed out over here. Basically this is eating and being eaten. But the most important, the main driver of every aquatic ecosystem is the primary production by phytoplankton, a process by which solar energy and CO2 is used producing sugars and oxygen, which drive the rest of the food web. Apart from solar energy, of course, also nutrients are important. And in the case of nutrients, there can be too little of them, in which case there is no productivity at all. There's an optimum range, and there can be very high levels of nutrients, which leads to all kinds of problems as shown in these photographs, which is actually one of the main reasons why phytoplankton models are very often employed and receive so much interest. So how do we deal with this in our Delft3D modeling system? To do this, we employ a methodology which is called the effect chain method, and it's shown in this scheme. In the middle part, again, is the natural system, and this time it's organized from left to right, from the physics up to the higher ecology, the birds, the ground-dwelling fish, etc. On top, there's a layer which indicates the influences that people have on the aquatic ecosystem, like nutrient policies, climate, and fisheries. The lower part of this figure indicates how all of this is dealt with in our modeling system. And we have chosen not to do this in one big overall global model, but we employ a system of coupled models. And they are linked, closely linked to each other, and they pass on information from one level to the other. The part we're going to talk about today is the part indicated over here. So the phytoplankton with all its with nutrients, with temperature, etc. And this is dealt with by the modeling parts called BAC and ECO or Bloom. But this there's also, this can only be done when there is information coming from here, from the waves, from the flow, and the suspended matter. So they drive this system, and it's also good to indicate that the eco model also has modules for zooplankton and benthos, shellfish, but we're not going to talk about these today. For historic reasons, you may find very many different names circulating in papers and reports, such as DBS, which is freshwater mode, GEM, the marine mode, ECO, DVAC, Delvac Bloom, and if you look carefully, you'll find even many more different names, which may be confusing, but the thing to keep in mind is that actually there's only one generic code, Delvac Bloom, and that's applied for all marine and freshwater applications. These applications only differ basically in the processes that are included in the models and the coefficient values, particularly with respect to the definition of the phytoplankton species. I will now tell you some of the big highlights of this model, which make it also outstanding in comparison to other models. First of all, it is a true multi-species competition model based on an easy to understand biological principle. Second, phytoplankton species are represented by different types, which allows Bloom to vary their characteristics as a function of the environment. Then, Bloom has been applied and validated worldwide in a very large number of fresh, transitional, and marine waters. Next, the result is a huge knowledge database with default coefficients available to every user of the model. Then the fifth point, Bloom typically operates on a 24-hour time step, which is characteristic for phytoplankton, but it does include diurnal variations in light, even across vertical layers. Then there's one additional highlight that I do want to mention because it's important, but will not be dealt with explicitly during this webinar. That's number 6. The mathematical method employed in the model produces numerical oscillations or instabilities under all circumstances. And any of you who have worked with other phytoplankton model will sometime have had problems with numerical problems like overshoots or undershoots or negative biomasses. Forget about all of this, it will never happen in Bloom. So let's look more closely to the competition principle. Bloom is a multi-species phytoplankton model, and there are lots of species as shown in this picture. Competition between phytoplankton types is the guiding principle in Bloom. Bloom selects the optimum composition, and it does this based on the ratio of two characteristics. First, the net growth rate, and second, the requirement for each environmental resource. And there is a trade-off principle between growth and requirement. Relatively high potential growth rates may compensate a relatively large requirement. So, opportunistic species win when light is high, but efficient species win when there is little light. Bloom considers various, something between 3 and up to 20 algal groups, including some nuisance algae such as Phaeocystis, Microcystis, or Ulva, sea lettuce. Every group, every species has its own growth response to the light conditions, as is shown in the graph to the right, growth response to temperature, growth response to available nutrients, and its own stoichiometry, that is the chemical composition in terms of carbon, nitrogen, phosphate, and chlorophyll. And all of this is depending on the environmental conditions. So what about these types? Why are they important and how do we deal with those in the model? Variability of stoichiometry is modeled as a distinction of the species in 3 different types: a nitrogen limited type, a phosphorus limited type, and an energy limited type. And you can regard those types as representative for the state a species will be in under prolonged conditions of limitation of this particular factor. Every type has a constant stoichiometry, and the model computes the optimum combination of the types. Different types of the same species may be present at the same time, and any linear combination of types is possible, so there is variable stoichiometry. Types can instantaneously convert into each other, but groups of species can't. So adaptation occurs rapidly, but succession, the replacement of one species by another, takes much more time. It takes days or even weeks. I will now tell something about energy, so light. At sufficient, that is not limiting availability of nutrients, ultimately light and temperature will become the limiting factors. And in analogy to nutrients, the available amount of light is calculated in the model. And a critical light intensity, light efficiency is reached when growth averaged over depth equals all the losses, which is shown in this graph over here, saying that the maximum growth rate, which is a function of the temperature, multiplied by the light efficiency equals the mortality plus the respiration, both of which are functions of temperature as well. This threshold holds for every species, and it varies in time and in space. So what does this mean? To illustrate this, I've included a graph over here showing on the x-axis the depth and on the y-axis the growth rate or the loss rates. So the blue line is the growth rate and the green line is mortality plus respiration. When there's a low extinction coefficient, so a lot of light is available, the amount of the depth average growth rate, which is the surface beneath the blue line, is bigger than the surface beneath the green line, and so there is a positive net growth rate and a surplus of energy. Now, as the turbidity increases, so the extinction gets larger, we reach a critical extinction level, which is exactly the number, which is exactly what we've shown in this relation previously, and so it means that the surface below the blue line is exactly equal to the surface below the green line. So this is called energy limitation, and of course, if the extinction gets higher, so the water becomes more turbid, then the growth rate becomes even smaller, and it's now too small to support to compensate all the losses due to respiration and mortality, and so there's a negative net growth rate and energy shortage. Notice that under stratified conditions, the depths get smaller. So we shift to the left, which means actually that there will be a higher potential biomass. As I mentioned, in Bloom, we look at a 24-hour time step, and I'll tell you some more about how this works in this model. So the first aspect to consider is time averaging. Growth, which is biomass increase, has a characteristic time of step of 24 hours. But notice that growth is not the same as photosynthesis, because photosynthesis is the production of energy, which is stored as sugar, and photosynthesis reacts quickly to changes in light. So you can say photosynthesis is an instantaneous process, whereas growth is a time-averaged process. And experimentally, it has also been shown that the light response curve of photosynthesis and growth are not the same. So apart from computational aspects, because a longer time step means the model runs quicker, there is also a fundamental argument to use a relatively long one-day time step as a default for Bloom. The next point to consider is depth averaging, because light varies with depth. In case of vertically averaged models, such as 2D models, the situation is actually quite simple, because the mixing depth of the phytoplankton equals the actual physical depth of the model, and that can of the segment, and that can easily be dealt with. Things get more complicated in 1DV and 3D models, because in those usually the mixing depth will not equal to the depth of the segment. In plain English, this means that during a 24-hour period, algae usually visit different vertical layers in the model. This conflicts with Delwak's usual procedure to compute process rates in segments independently from other segments during a time step of delta t. To deal with this, tracers are introduced in every vertical layer at the start of the Bloom time step. And the probability distribution of these tracers is computed for each layer based on the vertical transports, and based on the mixing patterns of the tracers, the depth averaged light regime is computed. This shows an example for one tracer. It's released in layer 4 in a tropical reservoir, and this is a 7-layer model. So after 24 hours, it appears that the tracer the algae has spent about 50% of its time indeed in the layer where it was released, in layer 4, but also a considerable portion of the time in layers above it, where the light intensity actually is higher, and some time in the layers below it where light intensities are smaller. Of course, in the model we do this not for one layer and one tracer, but we do this for all layers and all tracers simultaneously. And again, looking at one representative day, you get a picture like this, showing that, for instance, the tracer released in layer 1 spends about 50% of the amount of time in layer 1 and gets mixed up to layer 4 or 5 during this time step. Actually, this picture also shows that in this particular case, mixing in the upper part of the column is bigger than mixing in the lower part of the column, because tracers released in the lower layer don't get very much during the time step. To look at how this works out over a longer time period, this is a graph showing the result of all tracers in layer 1 in a tropical reservoir for a 3-year simulation. And there's a lot of variability in here, and which is important is that periods with little mixing, indicated here, are followed by periods with strong mixing. So this has a strong, clear effect on the production of the algae. But apart from this, also other factors play a role. And so the resulting vertical profile that you may compute with the model is also determined by these other factors. So this shows a representative series of plots for the depth profiles of algae in a deep region, somewhere in the North Sea. And it shows that in April, there is no vertical profile, so homogeneous mixing. In July, there is a peak of phytoplankton at the thermocline at 32 meters deep. In September, most of the algae are above the thermocline, and in October, there's homogeneous mixing again. Now, particularly, this picture is very interesting. And how can we explain this? It can be explained because in the upper part of the water, there's insufficient nutrients, but enough light because it's near the surface. Below the thermocline, there is insufficient light, but plenty of nutrients. And only at the thermocline, these conditions are optimal because there is both enough nutrients and enough light. The present tracer approach is confined to vertical excursions, which results in some inaccuracies, particularly in systems with tidal flats and deep lakes with confined shallow areas near beaches. We now have a beta model version considering both horizontal and vertical tracer movements, which will deal with this situation, and we expect this to be available in the coming months. So we will now take a bit closer look at how this competition principle actually works, because it's really important to understand this in order to understand how bloom works. So we start with a very simple case where we have two types and they compete for one nutrient. On the x-axis is the biomass of algae 1, and on the y-axis is the biomass of algae 2. And we look at one particular nutrient, dissolved inorganic nitrogen, which is the sum of nitrate and ammonia. And we notice that in this example, there's a difference in the requirement of these two algae. So algae 1 requires 0.07 per unit of biomass, and algae 2 requires only 0.05 per unit of biomass, which means that we can make a bigger biomass of algae 1 than of algae 2 than of algae 1 with the same available amount of nitrogen. So inside the green area over here, there is this reflects all the possible combinations of algae 1 and 2 where there is sufficient amount of nitrogen. On the orange line, this represents all the combinations of algae 1 and algae 2 where the nitrogen is 0. So this is called limiting. And outside the orange area, this represents all possible combinations of algae 1 and algae 2, which would require more nitrogen than is actually available. So in this case, there will be insufficient nutrients. Now we introduce the second criterion for the competition, that is the growth rate. And in this case, we assume that the growth rate of the two algal types is 0.5 per day. And inside this graph, this appears as a gray line, the one shown over here. And we start the competition experiment in the under the assumption that algae 1 and algae 2 are both 0. Then, if we look at we employ the two criteria at the same time, so the growth rate and the requirement. The first point to consider is the point shown over here, where we have algal 1 present, but obviously this is not the best possible condition. It's not yet optimum, because if we wait long enough during this competition experiment, then algal 2 will be the winner. And how can we check this? We can check this because, as I have mentioned, the competition criterion is growth divided by requirement, and for algal 2, the growth 0.5 divided by the requirement 0.05 exceeds 0.5 divided by 0.07. So algal 2, the one with the small nutrient requirement, wins. Now suppose we repeat this experiment, but this time algal 1 has a growth rate of 0.8 per day. Graphically, this means that the gray line has now shifted its angle. We also further assume that the nutrient requirement, the nitrogen requirement of the two algal remains the same. Again, we start our competition experiment at the origin. But now, if we look at we proceed with the competition experiment, we first end up in the point over here, which was the optimum solution in the previous example, but this case it isn't, because this point is now the optimum solution, and it is so because, again, employing our competition criterion, 0.5 divided by the requirement 0.05 is now smaller than 0.8 divided by 0.07. So algal 1 wins in this case, and it's the fastest growing, even though the potential biomass of algal 2 is bigger than the potential biomass of algal 1. To make things a bit more complicated and realistic, we now look at the competition experiment using two types and two nutrients. We assume that algal 1 requires less of nitrogen and algal 2 is more efficient in its usage of phosphorus. Again, the green line represents all possible combinations of these two types in which both PO4 and DIN are in ample supply. The orange line shown over here indicates nitrogen limitation, and the blue line represents phosphorus limitation. So in the other areas, either PO4 or DIN or both PO4 and DIN simultaneously are insufficient to support the amount of biomass that would be required to be in this area. Again, we make the assumption that the growth rate of the two types is the same. And again, we start our competition experiment in the origin. So algal 1 equals algal 2 equals 0. We start our competition and we end up, we first find a point in which algal 2 is present and it's limited by phosphate, but this is not the optimum yet. There's another point to consider with only algal 1 present, nitrogen limitation, but also this one isn't optimal. And this point over here is the optimum. And we now find that algal 1 is present and it's limited by nitrogen. Algal 2 is present and it's limited by phosphate, and so the two most efficient types win. And it's important to note that this correspondence between the presence of algae and the limiting factors is a characteristic of the model. You will always find exactly the same number of types as there are limiting factors, and you will always know which type is limited by which factor. So far, we have only looked at resource limitation, but what happens if we take time aspects into account? For instance, in this graph, this shows a situation where in the winter period there is hardly any biomass at all, but after some time there has been growth and a peak will be reached. So it takes about 2 weeks in this particular example to get growth started, and another 2 weeks to reach a nutrient-limited spring bloom. So 4 weeks are necessary. So how does Bloom deal with this? At the beginning of the growing season, algal 1 and algal 2 are both 0. Then, after some time, say 1 week, both algae have a biomass which is larger than 0, but obviously they're not yet resource limited because it takes time to get into the resource limited condition. This is dealt with in the model by taking the growth rate, the maximum biomass increase over time into account. So this line indicates the maximum growth of algal 2. This one indicates the maximum growth of algal 1 during, in this particular case, 1 week. And so after a week, we have both algae present and they are what is called growth limited. And again, as with resources, Bloom records growth and, by the way, also mortality limitations for individual species. After 3 weeks, biomasses of both algae have further increased, but there's still growth limitation. After 4 weeks, however, we find the same solution that we had previously. So now we have resource limitation, we have algal 1 present and limited by dissolved nitrogen, and we have algal 2 present and limited by phosphate. I will not show, but you can imagine that mortality limitations delimit the disappearance rate in a similar way to growth, because it also takes time to get rid of the entire biomass. Growth and mortality limitations also occur as a result of spatial gradients in depth, in nutrients, etc. This can be shown with this figure. So suppose we have 2 bodies of water, one is deep one and a shallow one, and in the deep one, algal 1 is the preferred species, and in the shallow area, algal 2 is the preferred species. If we wait long enough, then in the end, we will find algal 1 only here and algal 2 in this area. However, if we start mixing between these areas, then it's clear that mixing actually operates like time, because some algal of algal 2 will go to this area and some species of algal 1 will be moved to this area. So it extends the necessary, mixing extends the necessary period for reaching the resource limited condition. Similarly, heavy grazing by shellfish also results in prolonged periods of growth limitation. So when you actually run the model, you will find that there is often long periods of growth and mortality limitation due to these kinds of effects. In practice, Bloom of course considers all types and all limitations simultaneously, but this is more difficult to visualize. But keep in mind it works according to the same principle. We will now look at limitations and types in some practical situations. And we start at a coastal station in the North Sea, which is energy limited during the winter half year, which is shown over here. And accordingly, Bloom selects energy limited types, so Phaeocystus and diatom energy limited types in this particular example. Later in the year, in the summer half year, phosphorus and silicate are the main limitations. And accordingly, Bloom selects phosphorus limited types, shown over here. And usually, and that's one of the outstanding characteristics of Bloom, you'll find that many different types are present at the same time. So there is a very high species diversity. We now look at another station, a little further offshore, where conditions are different. Although in winter, again, energy is the main limitation. So also here we find energy limited types at the start of the bloom. But later in the summer period, now nitrogen is limited, silicate is limited, and at the end of the summer there's also some phosphate limitation. So again, in this case, we find in the summer half year nitrogen limited types to become dominant, and some phosphorus limited type, which is the one over here, at the same time. And there is less species diversity compared to the previous station that we looked at. One thing to point out in this particular case is that the blue one are the dinoflagellates N type, and the other one over here are the dinoflagellates P types. So this represents an example where two types of the same species coexist in the model. Also notice the strong increase in plankton C in summer, which is hardly reflected in an increase in chlorophyll, which is due to the intrinsic adjustment of the carbon to chlorophyll ratio in bloom. This is a type of phenomenon that is often observed and is lacking in many other models. Okay, I'll tell you something about the database with all the species. The freshwater model version consists of diatoms, microflagellates, green algae, Aphanizomenon, usually optionally as a nitrogen fixer, Microcystis oscillatoria, which is now usually called Planktotrix, Anabina, Nodularia, and it's been recently extended with Cylindrospermopsis, another nitrogen fixer, Pseudoanabena, and Gara, which are macroalgae attached to the sediment. The basic data set for the characteristics of these species have been obtained from work in the laboratory, plus literature sources, but there have been, of course, adjustments during model applications. The marine model version exists of a subset of diatoms, microflagellates, dinoflagellates, which can also be mixotrophic, which means they not only use solar energy, but they can also use organic matter as a source of energy. Phaeocystis, ulva, sea lettuce, which again is a macroalgae attached to the sediment, and benthic diatoms. And again, data from the literature adjusted during model applications. So in this big database that every user gets hold on when he uses the model are the stoichiometric ratios, so the nutrients, the chlorophyll component amount, the specific extinction coefficient, so the contribution of individual species to the decay of light, the maximum growth rate, the light requirement, the dependency on the diurnal light pattern, because algae react differently to variations in light intensity, the mortality rate as a function of the temperature and the salinity, The latter is particularly used to model transient conditions where marine and freshwater algae should be modeled simultaneously. The partitioning of mortality to autolysis, so dissolved nutrients, and to labile and refractory detritus, the respiration rate and the sedimentation rate. Bloom has been applied in a lot of cases, and I'll show you a very few examples of these. The first applications of the freshwater model version originate some 35 years ago, and the marine version is has a track record of something like 20 years now. Both model versions have been applied very extensively worldwide, initially mainly in the temperate regions, but many tropical and subtropical applications have been done in later years. The user selects the species to be included in the application, and usually the default coefficients of the species, which is part of the database, are sufficient. So there's very little need for local tuning. This picture, this figure shows the result of a 10-year simulation in a lake in the Netherlands, which used to be highly eutrophic. This is phosphorus, and phosphorus was strongly reduced in this lake, and so phosphorus went down, and this is simulated quite well by the model. The graph over here shows nitrate, and actually while phosphorus goes down, nitrate goes up, and again a very good model result. Here we show the simulation and observations on chlorophyll. While at the beginning chlorophyll levels are up to 400 or even 500, they drop to 50 or 100 at the end of this 10-year period. And again, this is very well reproduced by the model. And in response to the changes in the limiting factors, also the simulated species composition of the model changes from conditions where cyanobacteria are almost completely dominant to a condition where green algae and diatoms become the main sources. Does this fit into the measurements? Well, it does. This shows the result of 2 of the later years with respect to the individual species for which there were measurements. And although the pattern is not completely well reproduced like over here, in general, the overall picture is very good, and we notice that indeed this shift from cyanobacteria to dominance by diatoms and greens appeared in reality. Another example from a marine application is shown in this figure for a coastal station in the North Sea. The line indicates the model result, the dots the observations for the particular year, and the error bars indicate long-term 10-year trends in observations. This figure shows chlorophyll, which is a very good model result. Nitrate, again. For silicate, there's a little discrepancy in the spring, but in general, the result is also very good. And for phosphorus, there's something interesting because the simulation result of phosphorus measured very nicely reproduces the observations for the particular year, but both the simulation result as well as the observations of the later years are below the long-term range, which is the result of the fact that not only in the lakes but also the discharges of phosphate to the North Sea has been reduced in recent years. Here are two pictures of the species composition for diatoms and Phaeocystus. Again, there's a good model result. And you can also look at these results in a bit different way. This shows the amount of chlorophyll in March in a transect perpendicular to the Dutch coast, shown over here, from kilometer 0 to kilometer 370, and it shows that in this particular example biomass starts developing in two areas, one near shore where it's relatively shallow and one offshore where it's where the water is relatively clear. Yeah. Okay. This result shows the biomass of ulva, so sea lettuce, in Venice Lagoon. To the left simulated, to the right observed. Why are we interested in ulva? That is because it has caused problems in oxygen. And again, this shows the result of the model with respect to low oxygen conditions, and the dots indicate the measurements, and apart from some regions such as those over here, the model reproduces this pattern very well. Some results for the Sea of Marmora: chlorophyll in the upper layer, the intermediate layer, and the lower layer. And finally, something for a tropical reservoir: total nitrogen and chlorophyll. And of course, in the tropical reservoir, you may observe that the variation, the seasonal variation, is much smaller, but that doesn't mean that nothing is happening at all, because if you look at the vertical profile of the dominant species, Pseudoanabena, then it's obvious that both in horizontal direction as well as the vertical direction, there are strong gradients. So, to sum this up, Delft bloom is a phytoplankton competition model that has been developed and applied for over 35 years now. It's generic for both freshwater, marine, and transitional, so estuarine systems. It has many applications worldwide. It requires little time for calibration and is documented in a large number of peer-reviewed papers. And please keep in mind that water quality is often very much dependent on the hydrodynamics, so ideally the two should be modeled in close cooperation. Which brings me to my final slide, showing something for additional references, further readings. This is a list of reports and papers that I can highly recommend on the model, and they will indicate both, give you more information on the concepts of the model as well as a larger number of applications. Thank you.
And thank you, Hans. To our audience, thank you for attending this webinar. We hope you enjoyed this event. The webinar recordings and presentation slides will be available next week via both the Deltares Academy website and the Delft3D open source website. If you have any questions, comments or feedback, please contact academy@deltares.nl. For those who have questions for today's speaker, there is an opportunity to put your verbal questions to Hans Los after the live recorded broadcasting. Thank you again for participating and see you at our next webinar.